everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, episode 60 of Surfing the NASH Tsunami, our special coverage of the Madrigal Pharmaceuticals Phase 3 MAESTRO-NASH trial results, starts now.
Today, on this special episode of Surfing the NASH Tsunami.
Madrigal's positive top-line results certainly mean a lot for Madrigal, but they mean a lot to researchers, to patients, and also for a signal that liver health is open for investors and that it is worth the risk.
Yeah, if I were a competitor, I would be thrilled because with a 24% response rate, that leaves 76% of patients who need something else. Or something in addition. It's the best of both worlds from a drug development perspective.
If this really is as good as we're making it out to be today, we haven't even touched on the potential of reversal of liver cancer for these patients in the future.
There are systems that are self-amplifying, and you need moments when the system amplifies, but the rules change today.
On Monday, December 19th, 2022, Madrigal Pharmaceuticals made fatty liver history by announcing 12-month results of its Phase 3 pivotal MAESTRO-NASH trial. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and our guests, hepatology researchers and key opinion leaders Professors Scott Friedman, Mazen Noureddin, and Kris Kowdley, geneticist, age diagnostics CEO and entrepreneur Rachel Zayas, and patient advocates Donna Cryer, founder and CEO of the Global Liver Institute, and Michael Betel, founder of the Fatty Liver Alliance and lead of the Global Liver Action Network, as they discuss the impact of this pivotal moment in NASH history on their different stakeholder communities. Today, on this special episode of Surfing the NASH Tsunami.
Hi, it's Roger. Welcome to today's special episode. You can tell you've developed a rhythm with coworkers when you hear a statement or read a fact and know how your coworker will respond. Now, you know you've transcended rhythm when you go psychic, like this. Around 3:15 this morning, after a computer-free Sunday, I sat bolt upright in bed and said to myself, check your email. When I did, I found that Jorn had sent a note not even 5 minutes earlier saying, I think we should cover the Madrigal announcement. From there, it took 3 or 4 slightly groggy moments to learn what he was talking about and maybe 15 seconds to agree with him. So when I reawakened around 7 o'clock this morning, I sent notes to maybe 15 podcast friends and past surfers, checking to see who was free to record an episode today. 5 friends indicated they could be free at 3 o'clock Eastern, so there we were. But then our great buddy and former bandmate Donna Cryer indicated she could join us in the morning before a dental appointment. Happily, Louise was free at that time as well, so we asked Donna to join us at 11, added the interview with her before the 3 o'clock episode, and in the interim, Jorn, Louise, and I recorded the NASH Tag preview episode with Stephen Harrison. As you'll hear tomorrow, that one starts with conversation about the MAESTRO-NASH trial as well. At any rate, today we start with Donna, then we move on to the larger group conversation and close with an abbreviated business report. Today is really a special day for all of us in Nashville. This is the rising tide that lifts all ships. So sit back, listen, enjoy, and we'll all sail forth together.
For the first part of this episode, please join Donna, Louise, and Roger as they take a patient strategist perspective on the MAESTRO-NASH results.
Today, in honor of Madrigal, we are recording an episode at 3 o'clock, but one person we wanted with us couldn't make it at 3 o'clock. Eastern U.S. was Donna Cryer, who is with us now. Good morning, Donna, or good afternoon if you're Louise. How are you today?
I'm doing well. And for people who want to make sure that I am not ignoring all of my own health appointments, when does she fit them in? Well, the answer would be today. If the field would let me, I will be sharing some time with my dentist. And few people know how important dental health is to transplant recipients in particular, given that we're immunosuppressed. And so oral health and cardiac health are connected. And so liver health, of course, is connected to everything. So I am paying attention to all of those specialists who have felt slighted over the past year by my inattention. And so I am spending some quality time with them over the next few weeks.
I have to confess, I was at the dentist for an hour and a half last week as part of an ongoing procedure, but spending quality time is not necessarily the expression I would use.
But saying that, Roger, and following up on Donna's there, if you've got bad dental health, you've got a 6-fold increased risk of liver cancer. No figure because of the bad diet.
So, Louise, I'm proud to tell you that this is the first time in lots of years, like my entire adult life, that I've actually needed to have scaling done. So that might speak to some degree to my dental health. On the other hand, I have what they describe as burnished plaque. So they're doing opening my mouth a quadrant at a time instead of half at a time. And what I learned about myself last week from my dental hygienist is that I'm quoting now, I can officially describe myself as a badass because I get this stuff done with no injections. My wife, who gets her teeth drilled without injections, has kind of shamed me into it. But as a result, she said I can tell my grandkids that I'm tough.
I'm quite happy not to tell my future grandchildren that I'm that tough, to be fair.
As they say, it only hurts for a minute at a time. So, Donna, congratulations to all of us today, hey? Banner day for fatty liver disease.
I think it's so important when we think about the long-term sustainability of the field that Madrigal's positive top-line results certainly mean a lot for Madrigal, but they mean a lot to researchers, to patients, certainly, as we hope for an FDA-approved treatment in 23, and also for a signal that liver health is open for investors and that it is worth the risk. So this truly is the season of lights in liver health and in NASH. So I am happy to celebrate today, even though we know that there's much more work to be done.
So it's interesting. I think of you as being the person from the patient and advocacy space who lives on the higher plane, sees the entire field, and looks at the big picture. So I'm intrigued that you started with investors, in part because that's where I start. So you make me feel like I'm looking at this right. But why were investors the first place you went?
Because I am thinking about, and the Global Liver Institute is constructed in part to be a platform for sustainable innovation and engagement, identification of problems, and solving of problems in this greater liver health space. An individual company or an individual result or even an individual legislative win or education program, those are all good things. But I really judge the success of our efforts in advocacy. Have we made it a more welcome and a more productive and more efficient space for everyone to work? Investors, physicians, researchers, other advocates, other organizations who don't perhaps have a natural connection to the space, to companies who are in the brand and consumer space as well. I judge our success about the health of the overall field. And so to me, these individual results of Madrigal is certainly a result of their hard work, but it's also the result of lessons learned from other companies' experiences with regulatory, with the breadth of experience that Stephen Harrison brings from Summit Research, from all of the research that he does across the ecosystem and multiple molecules and products. And so I see this as representative of the health and progress of NASH as a whole, in addition to the individual success.
So, first of all, congratulations to Madrigal shareholders whose stock prices jumped 150% in the last day, from 70-something up to 213.51 at last sighting. So I think that's the smaller note. But do me a favor. I think you're the ideal person to do this. Let's pull on all the different threads that come out of this one, right? Because you just listed a bunch of them, but let's start pulling on them one at a time and see what it means for everybody for this to go forward. Investors directly was easy. We just did that.
Can I add, though, another one to that, investors? To female founders. I want to give a special shout out to Becky Taub as Chief Medical Officer and President, because we know that women attract a fraction, a very small single-digit fraction of the investment dollars that male-led companies get. This is a day of celebration for female founders and leaders in healthcare and everywhere else. So I wanted to add that.
Fantastic.
I think the next thread is the fact that they have, multiple trials. They are trying to answer multiple questions through multiple regulatory mechanisms. GLI stood our ground in the NASH Council about having a solution for every stage, where there were those in industry who wanted to pull us to say that only F4s or cirrhotics should be given focus, and we stood firm for years that there needed to be treatments for people across the NAFLD-NASH spectrum. And the fact that this research program has had success in F2s and F3s and has increased the evidence in even F1s and others. There are non-invasive as well as biopsy-proven NASH. The breadth of the program is, I feel seen, I feel heard, as they say, in terms of, leaving no patient behind, and really grateful hopeful that a company and their board and their investors and the research team would be willing to do the work to serve potentially a broad spectrum of patients, at least to develop the evidence further.
Yeah, I think, and kudos to the Madrigal team, and kudos to our former bandmate, Dr. Harrison, who's principal investigator, I think pushed a lot of that vision. Not alone, obviously, the company and everybody else had to be with it, but that's been his vision forever. I think he's probably having a pretty good day today, although I've not spoken to him yet this morning. Louise, questions, comments before we go on to pull another thread?
I think Donna did a great summary of that, and I think the only thing I would add to that is it was nice to see such a broad-reaching clinical trial that was just not focused on one specific way, but also looked at how this could be implemented in real world after and subsequently if the drug is approved. Because I think a lot of trials go in with we may not get approved, so let's just do this. Whereas this one appears to have gone in with we're going to get approved, we're going to be able to prove this, but how do we also get it into real world? Let's use all of those non-invasive therapies so that we cover a broad spectrum. That for me is a real strength of the data today, because I can see how this can be implemented through pathways depending on what you use. I am sure GLI have a very good plan of how they can legislate that and put that through, and how they can work through their workers and all of the people that they've supported within the field to advance liver care, because each one of them now becomes very vital as part of a hub and spoke. This is the education. We can now see how this does wherever the communities they've come from around the world. So that training program is absolutely valuable in days like this and over the next year of such excitement for patients. It's great.
I think that's a complete thought.
No, it was a complete thought. We're doing exactly what she said we'd do.
It's Christmas. I knew I was going to have one complete thought for the year.
Well, that was a rather exceptional gift to give the two of us. We like it. You gave a shout out to female investors. I also want to give a shout out to multiple founders, because I think part of the reason this program was so intelligently designed was because of the advocacy of the people they work with, but part of it is because for Becky and for Paul, this wasn't their first rodeo, as they say. And I think the first time you go through this and have to actually, to use a very female metaphor, if you will, birth a drug in the marketplace, you learn how many things can go wrong, and you probably get a lot smarter about what to plan for in advance than if you are truly an advocate but never been to the dance before. The breadth of this program and the intelligence with which it was designed gives testimony to the founders as individuals, to the people they worked with, and also to the experience you get doing this a couple of times.
I will quote George Clooney, who said, "I have learned nothing from my successes and everything from my failures." That is very true. And I am really impressed that this group collectively have the humility to learn from those failures and turn them into life lessons, into research lessons, regulatory lessons, team building, lessons. There are, many folks from the larger Madrigal corporate team who have been in other companies and environments and have learned various things. And so, I think of this as sort of the apotheosis of all the lessons learned by living life in the arena.
Amen to that.
Donna, do you see this data and the strength of it as moving us beyond the biopsy in everything other than a clinical trial to prove drug efficacy? Because the retention rate of these patients, I think there were 997 or 995, and only a very small set of patients did not make endpoints, and some were ruled out because their second biopsy or end of treatment biopsy was outside of the 10-week or 8-week window for COVID. They couldn't make it, but they still attended for those biopsies, so they weren't included in the data set. But the retention was massive despite the biopsy, but it has taken us beyond. Do you think that is exactly that sort of legacy that something like Madrigal's trial can give?
I hope it's not used against them, that there was such great retention, so that people won't say, see, patients don't mind biopsies. We do. It's just that the study team showed enough care and follow-up and engagement with their patients, and patients showed enough commitment to the trial and to the research to do this thing. It should not be used against the field that patients retained in a biopsy-determined trial. I think that the 17%, when I was starting to read through, who either did not continue in the trial or had their biopsies too late, if this was sort of a thriller, my heart stopped, the dramatic music would have played, and I was looking, would they still have the same effect size, even, with these patients. And the fact that they did just speaks to the strength of the compound and the program. But I think we need to look at, is that an appropriate way to characterize those patients in the trial design? And I'm encouraged that they are doing specifically a non-invasive trial to be able to develop that data as well. I really think that there are two factors. It's going to be up to, AASLD and EASL to have guidelines that do not include biopsy. And there are going to have then these trials that are submitted to the FDA and biomarkers, qualifications programs and things on the validation of non-invasive technologies, so that we don't end up with a label for any drug. We get so excited, we get to this point, there's an FDA approved treatment, but then it has biopsy limitations on the label. That would not be a great outcome. That would not be a success. That would not be a day for hurrah. That would mean extremely limited and restricted and lack of access to whatever medication is approved. It would point out this huge gap between the real world environment and the research environment. It would have implications for equity and accessibility. It would not be a success if we were restricted to biopsy in the label. So I maintain my focus and my concern in those areas.
So, first of all, Donna, before you came on, Louise and I had been noting that every conversation we've had this past week, except the one with you, wound up touching heavily on the World Cup. And I figured out how to get the World Cup into this conversation courtesy of your last comment, which is our colleague, Dr. Harrison, if you've listened to him on podcasts, which we all have, talked about Michael Jordan and how many times Michael Jordan missed the last shot of the game so that he could make the ones that he made. Louise, I don't know if you recall this, but right at the end of the 2014 World Cup final, Leo Messi had a perfectly makeable direct kick that could have tied the game and sent it into penalty kicks, and you saw the look on his face and you knew he wasn't going to make the kick. And I didn't see anything on Messi's face in 120 minutes plus penalties yesterday that looked anything like, I've been here before, I failed before, I'm not failing this time, I know how to get it right. So that's my World Cup reference for today, Donna.
My World Cup reference is that my personal trainer constructs my hour-long sessions with an increasing amount of soccer drills once he learned that I knew how to play. I think it was trapping and passing with my left foot that clued him into that when we were doing a warm-up drill. And so throughout the World Cups, more of my session has been in soccer or football for the global community, in football drills.
That was a hidden talent we didn't know.
And you've already done the one thing we wouldn't know about you if we didn't ask. So now you've got two, Donna.
I was a midfielder.
Becoming a true woman of mystery. So was my older daughter, by the way. My younger daughter was a keeper. My older daughter was a midfielder. And I was a midfielder. So, yeah, we all have that in common. So let's pull on the patient thread. What message do patients take out of this?
I hope that the message the patients take out of this is, one, that there's hope. Two, that the field has really dug deep. When you look at not only the liver effects, but the cardiovascular effects of this compound, that the attention paid to people's cardiovascular health as well as their liver health, has been rewarded. And so we're closer to a whole person approach for this. I think that the note for patient advocates is that we will need to be very ready for next year to make sure that this comes all the way across the line. I play more baseball than football, so running through the base is what I think about in my father's counsel, to run all the way through the base and not stop lest you get called out. And so to me, running through the base means testifying for an approval, helping with the education afterwards, working with payers to make sure that there's meaningful access and adoption. So we need to absolutely be ready for a coordinated and intense, very body of work and period of time where advocacy will never be more necessary than it is then to get this all off on the right foot.
We have an opportunity here with this early release and the big hope that this is going to fast track soon after obeticholic acid. If we get all of the systems right with Donna's lead and other patient advocacy groups to get this drug through the FDA and get the pathways right with payers, this means that potentially when the EMA come to approval, we already have the networks and the pathways in place that can be rolled out, and we can learn from each one for each of the unique healthcare systems. The NHS will be different to the French system. We know they're all funded differently. How it will affect Australia, who don't necessarily look at liver poor health or fatty liver disease with the lack of information, they're going to be slightly off guard here, but I think it's also a system that can upskill very, very quickly. So it will depend. We do have a unique opportunity to get these pathways in place because of the experience in hepatitis C DAAs and how that's been rolled out in the world quite successfully in different areas. So I think we take off our strengths, going back to what Donna said earlier, we learn from where we had disappointments and where it didn't work, and we make a better foundation going forward so that we are in the right place at the right time to be able to locate, treat, advise as a community, because we now have cardiology and endocrinology. And the nicest thing about this study was that those comorbid conditions were part of the protocol. Rebecca commented that there were 3 metabolic conditions included in this. So we are not looking at a drug just for NASH. We are looking for a drug for NASH in those other populations, which are now strengthening their guidelines with the American Association of Diabetes now publishing their quality of care metrics yesterday or for January, which include NAFLD.
I think that's an excellent point. And one of the things that goes along with that, which is somewhere between learning from everyone else's experiences and what you just described, was that the one secondary endpoint they called out in the press release today was LDL. 2 reasons to do that, right? Number one is because LDL is where OCA foundered. So it's a message to the investors where Donna started that you don't have to worry about that here. And then it's a message to the broader world that cardiovascular really matters for the patient populations we're talking about. This is the first thing people worry about, not to worry. So I thought that was a really powerful dual message to include in the initial statement today for the reason we started with and the one you just went to.
I frequently, as I travel throughout this space, this community and field, I think, what if physicians thought more like lawyers? And certainly, what if lawyers thought a little bit more like physicians sometimes as I try to bridge the 2 communities. But when I see this work done and the press release, I think, oh, here's some physicians or at least PR people who thought like lawyers. And what I mean by that is anticipating the questions that you're going to be asked and answering them proactively, showing that you have evidence that anticipates and answers those questions. And so one of the reasons why I give them an A on this test is that they did just what you said. They anticipated based on what has gone on before rather than thinking that they're dropping this into a vacuum. But in the context of what has happened, what has been the clinical and regulatory precedents, they have anticipated and answered the major questions. And so it makes them look that much more thoughtful and prepared, and they bring then a lot of credibility into any process or conversation in which they would engage.
It was interesting. Stephen, when he was speaking, actually used the comment, treat the person, not the disease, as part of the thing. And we use that a lot on the podcast, to treat the person. You're right. It was a particular skill set. They've thought all around 360 degrees and done a very good job on most of those degrees. So yeah, hats off. And I think Rachel Zayas interviewed Rebecca Taub when I was with her at the NASH Summit when we were in Heathrow. That was earlier on in the year, and I'm sure she had a lot of information then.
Actually, Rachel's joining us for the 3 o'clock podcast, in part because I want to talk with her about having interviewed Becky Taub and a couple of other reasons to have Rachel there, but one of them is specifically to ask about that interview and what she learned in that interview that informed what she heard today.
One question I would have for, and I just throw this up for thought and conversation, what effect would changing the name have on this momentum? Does it add value, positive value to this process, or does it detract? I won't give my opinion. I think it's in my tone.
If our colleague, Ms. Campbell, looks like the cat that just ate the canary, it's because our discussion right before this was with Jeff Lazarus, and when reviewing the nomenclature conference, that was exactly her question. And yes, that question will show up this afternoon again for various opinions.
Great. And I think you should, in your social engagement, that may be the question that you put forward to people throughout this process. However, people have tried to present it. My participation has been how to make sure that this process is not just an academic exercise, that the real world implications of any changes are taken into account and are in fact research documented and accounted for prior to any change. And I think that's what leadership means. I think that's what the field, whether patients, industry partners, investors, I think it's what the field is owed by anyone contemplating such such a change at such a time as this. So I wonder how those who feel that this is about publications and impact points feel their contribution is to this process at this time.
If I had to guess, based on what we heard this morning, and Louise, if you think I'm not representing what we heard this morning accurately, tell me, it would be that the pressures to do that are broader, but boy, we better do it now because there can't be any confusion when the drugs get approved. I think that's what we heard this morning.
That was true, but I thought there was also a recognition that gathering momentum could be positive even if there was a name change by publication and making people aware of it who'd never been aware of it and using that as an opportunity. Yes, if we change the name, you need to know the new name. No, if we don't change the name. So I think it was a good grasp of were there opportunities in a name change, whether or not it goes down well or not. And I think that was the addition which I wasn't expecting.
Fair. And for that reason, do it now, right? Whatever you're going to do it quickly because you can't have any confusion left by the time you get there, whatever the outcome is.
Right. So, I mean, we'll make the most of whatever is. We just need to don't pretend like it's not having an impact, and we don't have to do work to mitigate any of the negative effects. Don't pretend like it's just a thing that doesn't have any real world implications, that won't put some things back and potentially, if we work hard enough to make them so, move them forward. Don't pretend that it won't be work. That's all I ask of people.
I think that's fair. Look, I've made the comment before, I think with you and without you, that one of the striking cultural issues around this one is the closer you are to dealing with the commercialization of medications in advanced economies, your position on this starts to change. The further away you are, the less it matters. The closer you get, the more it matters. And I think that's right. I think one of the things that happened today is we moved a lot closer. Hey, demonstrably closer today. Any other thoughts to share with us before you head off to the dentist and Louise cooks dinner and I get ready for our podcast in 2.5 hours?
I'm going to look forward to listening to the other voices. I want to be so thankful for you for making this extra opportunity today. We'll continue to sift through the implications of this and the multiple trials and to watch it. But I look forward to hearing other people's hot takes of this. And I just thank you again for creating this multifaceted platform for these informed views so that the rest of the community can have access to them as well. So thank you.
Thanks. It's a great thing to get to do for us. We have fun with it. It's a way to make a contribution. And this is a great day. So with that, let us send everybody off to their respective personal tasks. And some of us will reconvene in a couple hours and some of us won't. Thanks, folks. See you later. And with no further ado, here's our group conversation.
Next, please join Jörn, Louise, Roger, and their other guests for a far-reaching exchange on the MAESTRO-NASH trial and its implications for the various stakeholders.
So a small personal vignette. It's 3:00 in the morning. I didn't look at any email or internet yesterday. I try not to do that one day a week if I can. And it's 3:00 in the morning, and I sit up in bed and I say, I better go read my mail. And I do go read my mail. And what's there but a 5-minute earlier note from Jörn saying, I hope we're covering the Madrigal announcement today. At which point I said, well, gee, Madrigal announcement, I better check the internet. And so all this has been put together since then because Jörn figured out how not only send me an email, but to get it telepathically into my brain as I slept. Do you know that you have that power? Or is that the first time you've ever been able to demonstrate that you can prove?
No, I'm sorry I made you wake up in the middle of the night, but I'm glad you pulled this great crew together to get different views on it. And I have to say, I had a big excitement all of this day, and I will see how my colleagues and respected friends are feeling about this.
So we also have 8 people on this podcast, which we've never done before because we wanted to get a whole bunch of different perspectives. So we have Jörn, we have Louise, as usual. Louise, welcome back for the, I guess, 3rd or 4th time today, eh?
Yeah, long time no see.
Yeah, you've been around for most of it. And then we have, I'm going to do this in order on my screen, going from the top right. We have Rachel Zayas, our good friend and entrepreneur. Rachel, how are you?
I'm doing really well. Doing really well, thank you.
She has shared that she is in a chilly space today, which will keep her on her toes the whole conversation. We have Mike Betel from Fatty Liver Alliance and the Liver Action Network. Mike, how are you?
Great, thank you. Excited to be here.
And then going around down to the bottom, we have Kris Kowdley. Kris, how are you this afternoon?
Doing well, thank you.
Good. Exciting day for all of us. And Scott Friedman. Scott, how are you?
I'm terrific.
And finally, Mazen Noureddin. Mazen, how are you today?
How are you guys doing? Glad to be here.
Fine. On our last conversation with Stephen previewing NASH, Louise did make
make the comment, which I think she just shared with you before we got on, that for one of your presentations, she's guessing you're going to have to change a whole bunch of slides because of what happened today, because what's going to happen before that. She also speculated that if the Resmetirom is the first of the 3 late drug presentations, that there are going to be so many questions on that one that we're going to wind up an hour behind before we get started. But I'm guessing that the moderators can control that one. All right. So we're here on short notice, as I noted already, because of this morning's announcement from Madrigal Pharmaceuticals. I'm going to use a different groundbreaker, which is I'm going to ask everybody to share a reaction you had in terms of how that announcement affects what you do every day of your life in connection with fatty liver space. Bold one, go first.
I'll jump in if that's okay. This is so important because it reopens the floodgates of interest by all stakeholders in treating this disease, not just the investment community, but patients, providers, everybody who was a bit fatigued by the negative results that have been piling up and the concern that maybe this is a disease that can't be treated, although I never believed that. But just the idea that the space is alive and well and ready for continued growth and advances.
It's a great start.
I was like, oh, finally. There's no disappointment. Quite happy. It's, as Scott said, it's great news. It tells us a lot of things that we have learned throughout the years. It still opens the door for more work for all of us. It talks about combination therapy. It talks about that we can reach efficacy. We'll talk about details, but there was some consistency with the previous trials. And I want to congratulate the patients first, Madrigal second, NASH researchers, and many people around the world for this. I think this is a great start for first phase 3 drug to be approved, hopefully.
I was incredibly excited. Actually, I found out from Roger because he asked if we were available today, and that's how I knew about it. And then I immediately went online and started to read all the news that came out. And so for patients, like Mazen said, the patients that have been waiting so patiently to have something to be able to treat their NASH is a huge step forward. And then at the same time, the other side of the coin is, are we ready? And are, for example, primary care physicians ready for all of the huge number of people that are going to be coming forward when they start to hear the news? So exciting day.
Yeah, I would echo all of those comments. I think there are several other key points that should be emphasized. The fact that there was a dose-response relationship between efficacy results speaks to the activity of the compound. The safety is very reassuring. The additional favorable effect on LDL cholesterol is another finding that, I think is very encouraging to us. It's interesting that the results in terms of fibrosis are similar to what we've seen with the other positive phase 3 study. But the favorable effects on lipids is certainly something that is even more encouraging in the sense that we might have metabolic and favorable lipid changes as well. And to achieve both NASH resolution at a fairly attractive rate compared to placebo, really a threefold improvement over placebo, along with fibrosis reduction by one stage in this trial, I think is very encouraging. But at a broader level, I think in terms of drug development for NASH, this shows a path forward. I think we still need to continue to challenge ourselves and the community on developing non-invasive tests because they paralleled the histologic findings in this study. And finally, I would say that the idea that, we can see successful results now in our second phase 3 trial, I think is very much a shot in the arm for those of us who really believe that there's a huge unmet need and, we need different types of pathways to be explored for treating this epidemic at this point.
Yeah, if I were a competitor, I would be thrilled because, number one, this is positive. Barring something unforeseen, one would hope and expect the drug will be approved. But with a 24% response rate, that leaves 76% of patients who need something else. or something in addition. And so it's sort of it's a best of both worlds from a drug development perspective. Certainly, we'd love a home run and pack up our bags and move on to something else, but that's hardly the case. We need to do better in capturing benefit for all patients, not for 24%. But this is a major step in the right direction, and it reinforces to everybody that this is a fight worth fighting because we can move the needle.
Yeah, it's great. I had two thoughts, also on the reflection of the colleagues here. The first thought is this is the second positive phase 3 trial, and the first one had some troubles crossing the finish line based on the side effect profile. And Kris mentioned this. I think we're not seeing that same deleterious side effect profile here. So I was wondering, there can't be the same bothersome progress in FDA negotiations here to not get conditional approval, because really I think that's been cleared with that results. I mean, we're seeing not all, these are top line results, we're seeing not all the details, particularly on safety, but it doesn't sound like safety is going to be in its way. So that's great news. The second thought I had was, as Kris mentioned, both endpoints are met. The EMA actually requires, in contrast to the FDA, both endpoints to be met, which to me felt, well, even European agencies might consider this drug to be approvable, which is, great news for us on this side of the world.
Can I add also something on the safety? I just want to remind everyone that there were data presented also from their open arm, single cirrhosis arm that relied on non-invasive testing, and the drug was safe in that cohort in a previous presentation. So I think we have a good profile of safety data. And there are many comments actually to be made about this study, and there are many lessons to be learned. So happy to keep going with this conversation.
So for most on this call, you know that I am new to the NASH space. We pivoted the company 2 years ago from hepatocellular carcinoma to focus on NASH and fibrosis, and many said that this was a mistake because there was no therapeutic agents that had been approved, so there was nothing for patients to do except have lifestyle modifications. And I pushed back on that, and I said that those comments are betting against science and betting against the scientific method. So really excited to be part of this conversation today. And I think, as others have outlined on this call so far, this changes the narrative for various key stakeholders, for patients, providers, for other drug developers, as well as those developing medical devices, specifically non-invasive diagnostic tools. So this changes my day-to-day life and my day-to-day operations, because this is the second positive readout. And this is exciting and innovative. And I think that this is really going to change the strategy going forward for so many.
Unless we think this was a shot in the dark, I think there's one statistic that's very impressive, and that's a 10% placebo response, which is far lower than other trials, and I would only imagine reflects the accumulated wisdom of the field in enriching the population for patients who need the drug and are more likely to benefit from it. So hats off to people like Mazen and Kris and Jörn, who are spearheading a lot of these clinical trials.
Just to comment on that quickly, Roger, then I'll help back to you. Be reminded that the trial actually required 3 metabolic risk factors, elevated TE, to be present in order to be screened. So I think, for those people who wonder, is this by pure chance that we're seeing this now? I think the response is this is a more targeted selection of patients. Very simple, 3 metabolic risk factors, transient elastography, then biopsy to get enrolled. So I think that's an important point.
I want to add to that, actually. Those are very, very important comments. And it started hearing people comparing this to this drug and that drug. And I want to caution people about comparing to other drugs, because I think the placebo, I hate to say it, I called it a little bit bipolar this morning, but the trend in these trials are as following. As Scott said, it's one of the lowest placebo response we have seen. Nevertheless, phase 3 studies in general, they seem to have a lower placebo response compared to phase 2. So the New England Journal papers that came out from phase 2s, to Scott's point, they put NASH resolution at 17-18% and fibrosis improvement and between 16 and 20%. Now, with the obeticholic acid data, they actually had also low NASH resolution for placebo, and they had low fibrosis improvement placebo, similar to the Madrigal. Indeed, the Resmetirom data, the placebo response was slightly higher than obeticholic acid, which speaks of also the robustness of the results. So if I want to recap it, phase 2 studies seem to have higher placebo response compared to the phase 3 studies. Now, another point actually, which if you look at the Resmetirom previous trial, the NASH resolution was about the same for the previous study to this study. So you can argue that data held consistent. I think they were in the 24% then, and now we're at 30%, but their placebo was lower in the phase 2 study. I think the NASH resolution was about 6 to 7%. They did not achieve fibrosis, and the study was not powered to achieve fibrosis in their phase 2 study, but their fibrosis and placebo fibrosis improvements was in the 20% back then. So the good news is they held consistent. They're showing that NASH activity resolution lead to fibrosis improvement, which I actually did not bet on for this study. I said they will achieve NASH resolution, not fibrosis improvement. So they hit both, which is great. And I think, as to Scott's point, we're learning in the field and things are improving. And I want to remind the audience that this trial had 2 pathologists reading these slides.
Duly noted about the pathologist reading and all the other points. I just want to share a funny comment, which is when we talked to Stephen Harrison a little earlier today about this in advance of the episode about the NASH preview, he started by talking about what this meant for the patients. Mazen started by talking about what this meant for the patients. When we talked to Donna Cryer earlier in the day, she started by talking about what it meant for the investors, which I thought was interesting but not inappropriate, because ultimately what investment will benefit patients really more than anybody else, right? Think about Mike's comment about the need to educate patients and frontline treaters. We can't do that without money, and money has been in short supply. So the investment will not only fuel the drug development, which is critical, but it will also fuel the education and marketing support and the other things that will really speed the diffusion of information. One of the things I like about this kind of thing is it really is a web, right? So many different places get touched. And then also to Rachel's point, one of the reasons I wanted to have Rachel on today specifically is because I wanted to ask Rachel a question we'll go back to in a couple of minutes, but which is, how do this collateral entrepreneurs specifically benefit? I mean, we know what this will mean for the drug developers, and we know what it'll mean for the big device and diagnostic companies, but what does it mean for the people who are really trying to move the envelope out at the margins, kind of doing the work that Rachel's doing? Since the hope is that in 10 to 15 years, it's work like what Rachel's doing. And Rachel, it doesn't mean you're going to have to be in the wilderness for 10 to 15 years. But the hope is that in 10 to 15 years, the kind of work that she's doing right now is going to be what pushes us way further ahead, what she's commercializing. So there's this real weave as we dive into the study, which is important. But to keep in scope, how many different stakeholders stand to benefit from getting a successful trial over the line and getting it to FDA approval? Louise, I guess you're the only one we've not heard from yet. What are we missing?
I agree with everybody that's had their input on it. And I was thinking I was going to start off with where Jörn was, that the breadth of the comorbid conditions and metabolic diseases included in this trial just means that it ticks a whole load of different boxes when we talk about liver and poor liver health potentially in the future. But I think for me, when I think about interactions with patients now, we can talk about, if diet and lifestyle does not work for you, we have another option. We can hold and we can try diet and lifestyle. But actually, if it isn't successful, there is something else. And that is always a massive incentive, that if you don't achieve the lifestyle and diet that's required, you're not a failure. And I think so much of the time we do focus on lifestyle, we do focus on those cornerstones, but it is really hard in primary care to get people to do that when it's the only thing you've got to continue to do. So by having these next steps and the encouragement in the field, I just think is such a great step for the patients, and to thank every single patient, not only who's done Madrigal, but every single patient that's ever done a NASH study has contributed to what was released today that changes the future for every further person in the NASH and NAFLD space, and a lot of the comorbid metabolic conditions, because we've seen those safety profiles, which is highly encouraging.
I just want to agree, but I also want to bring up another angle, which is the following. I think diet and exercise are absolutely the first line of therapy, as well as should be the therapy all the time while you're taking drugs. Here, I don't want to say, well, by weight loss, and I just want to make sure we're not saying try weight loss and exercise, and then we have a drug for you to try if you're not responding. The reason why this trial included actually more F3s than F2s patients. I think they were in the 60% or so. And thus, I would argue that if you have the patient in the first visit in your office, he will say, you need weight loss and exercise, and sit down with them and tell them about weight loss and exercise is better if you have a nutritionist, because I'm sure none of us will spend enough time on that. But at the same time, if you have an F3 patient, I would argue a drug should be started in that first visit or at least the second visit. Another thing also I liked about this study, in addition to the metabolic risk factors that Jörn mentioned, they had also about 20% or so Hispanics, and this is a little bit on the higher side from what we have seen previously. And this is encouraging because it started looking at the patients at high risk. So there are a lot of positive things that need to be looked at. The last point I also want to make, I want to go back to Kris' analogy of looking at the drug and its efficacy. He said threefolds more for NASH resolution, and I do agree with that. It was almost twofolds for fibrosis improvement, rather than looking at the absolute numbers across trials. This is how I want to look at it, because the histology is all over the place. So I think this is the right approach that Kris started.
To follow up on that and feedback, get maybe feedback from Kris, who's done a lot of drug development in his career, is something that was said in the press release was today. Well, we're seeing 2 effective doses, so we're taking 2 doses forward. And, we're looking at top-line data, so there's a little bit more efficacy, a little bit more side effects versus a little less efficacy, a little less side effects. If it's one drug, do we need those 2 doses? And not sure anybody has a thought on that.
Yeah, I'll jump in. I do think that it's useful to have this information because, first of all, we've gotten pretty choreographed into standard approaches with regard to enrolling patients in trials. F2, F3 fibrosis, compensated liver disease, platelet counts are normal, all liver functions clearly normal. As we move forward and we get a better understanding of the indirect measures of activity of thyroid hormone beta receptor agonists with regard to sex hormone binding globulin or other features, we're going to want the flexibility for patients with a broader spectrum of liver disease, because I think, would like to have the option of knowing that there's some efficacy that can be offset by potentially tolerability or clearance of the drug or some other factor that would allow it to be used in a broader range of patients. The other point I think is important is that, as we know, who are in the trenches and take care of a lot of patients, every patient is different. So there is a dose-response relationship in terms of adverse events that was reported. And some patients may have tolerability issues that might require step-down therapy. So knowing that you don't have to sacrifice efficacy if the patient is not able to tolerate the optimum dose, it gives us as clinicians flexibility. So I think both having data that shows efficacy in a broader range of patients that might potentially be better suited for a lower dose, and also, more importantly, show that tolerability can be affected variably in different patients, but they can step down and still get efficacy, I think provides useful information. So at this point, it's sort of a best case scenario, if you will, where you have efficacy shown with both doses, and what trade-off you might have in terms of efficacy, maybe you'll pick up in terms of tolerability.
On that point, Scott was saying earlier, it's a welcome result for everybody else because there were 76% of patients that didn't. So drilling down into who did not succeed. Yes, I was really excited to see a 21% Hispanic, but if they are the percentage that didn't respond, then we're looking at targeting the right populations. If we look at the average BMI, I think it was 36. Is the lower dose better in those with a BMI between 30 and 32, for example? Drilling down in who did, but more importantly, drilling down in who did not respond in these tests, for me, is important, because then we become more specific of who we're going to treat in the future, the same as we were in the same way with genotypes 1 to 4, different things target different people. The wealth of data that we can get out of this for me is just phenomenal. And it's going to take years, but I'm excited about those years coming.
If I can jump in, I completely agree, and to put a fine point on it, what are the genetic, if any, makeup or the histologic or the molecular features of those who responded versus not? And of course, now we have some new problems, which is, are we going to be able to convince payers to pay for it? Number one. Number two, since it didn't work in most patients, how do we identify those patients, get them off the drug if we can't pre-select them so that they can get onto something else and maybe move more towards personalized therapy? And the final implication, or one other implication, is, of course, that clinical trials going forward will no longer be placebo-controlled, I would imagine, unless they're either going to be patients who are being compared to Resmetirom or in combination with Resmetirom and another drug, or have some, which I can't think of, some contraindication to using Resmetirom, but can get a different drug. So this really shifts the whole ecosystem considerably from those perspectives as well. And obviously, we have a lot to learn.
I really like the precision medicine that Scott mentioned. I'm asking a question. I'm not sure if Resmetirom will become a standard of care for trials if it's a conditional approval versus the placebo continue. I don't know what's the right answer.
Well, it's possible. I don't know either. You guys might know better than me.
But also, I think the opposite of precision medicine, and I like, again, precision medicine more. The question is, we have to be reminded this is a chronic disease that took many years to take in place. So this is a 52 weeks trial. And I want to be flexible in terms of looking for responders, non-responders. Would a longer duration lead to a better improvement? Another thing is, I really think the field is going to be not a drug better than the other. Rather, you want to add, for instance, the GLP-1 to TH-beta receptor and see how they do together, which is to Scott's point, that's probably going to be compared in the future. So a little bit patient with the responders versus non-responder analysis, and let's see what it does in the long term and how adding other medications will lead to additional, even synergistic.
I wanted a couple of minutes ago to comment just on what everybody was saying. The tailored therapy approach is going to be the future for sure, and the subset analysis of the data will help us get there. There were some, Louise mentioned the diet and exercise, which of course everybody talks about. There's also gastric balloon and gastric bypass. So, at which point do some of these other options come into play? So there's the pharmacological option probably for the masses, but we're going to have to tailor, probably based on the data, which treatment option is best for that patient.
One of the things I find really exciting, and this conversation takes me there, and particularly Mazen's point, they made the point that this is a 52-week conditional endpoint in a 54-month outcome study. So we're going to be looking at these patients for a lot longer, this set of patients for a lot longer even than what's being reported today. And they also made a point of discussing the other 4 trials that are going on in parallel, right? So the amount of data that we're going to be looking at on Resmetirom as we go forward is really compelling in both its breadth and its length. When you lay that alongside obeticholic acid and the idea that we've already got patients who've been on that for more than 4 years because they continued their trials, we're starting to see the emergence of, I think, a much richer and multi-dimensional sets of data, if you will, that will allow research for years, but also will allow fertile minds to go in just a ton of different directions, which I find really exciting.
The other point that I think is important to emphasize is not only what this means for patients, the investor community, drug developers, and others, is just in terms of moving the science forward. I mean, we had a lot of information about FXR agonists and why they might be effective in this particular condition. For a long time. The concept that a, selective thyroid hormone beta receptor agonists might potentially make the liver metabolically more efficient, maybe improve not just beta oxidation, but fat disposal, de novo lipogenesis, many other factors. But just in terms of the science of understanding how functionally hypothyroid many patients with NASH are, just as we've now learning more and more about how they tend to have sarcopenia that is not recognized and not suspected for a long time, is a huge advance. So this was in some ways a proof of concept. We had a lot of preliminary data suggesting that there was a good likelihood that the results would be positive. But in terms of now reversing back and reverse engineering, understanding the science, I think from a translational perspective, there will be huge opportunities. And I think this will have implications for patients even who are not on treatment at the present time in terms of how do we better investigate thyroid function in the liver's sensitivity to thyroid hormone, and the downstream effects of thyroid hormone receptor agonism in this condition and other conditions that may be adjacent to NASH, if you will.
To build on Kris' comments he just said and earlier, and what you said, Roger, we're going to have a trial that is ongoing for years. So the LDL effect, I do wonder if it's going to continue over time, and we're going to start seeing MACE favorable effects. And I hope that will be the case. So here, like you're starting addressing the disease as a whole body rather, or at least from two big comorbidity standpoint. And I hope that will be the case. So there's an excitement with the LDL data as well.
That is true. And that's something that wasn't discussed yet from us. I thought, one thought I had, what does this trial mean for NITs? Because it's particularly rich in non-invasive tests, and we're seeing response. And the question came up, who does respond? So, the take on, we're going to see a lot of compendium diagnostics, I think, here, which might be qualified, hopefully.
Yeah, but if you remember a lot of, the GLP-1 data, they do studies on multiple populations, as you said. Some of them, like they don't have cardiovascular risk factors to start with, and they see if there are any effects. And some of them, they did have cardiovascular comorbidities, and if they see if there's any effect. So your point is well taken, actually. I do wonder if we'll see anything, but it's just encouraging to see such a data. And if there's a signal at the end of like 4 or 5 years, I'll be very excited about it. And once we get through the liver biopsy, we can do 5 to 10,000 cohort data and look at MACE and non-MACE and the points that we wish.
It may well be that the FDA at some point will require them because, as I mentioned, nobody wants the patient to remain on a drug that isn't working. Most of all the patients, but also the payers. So a lot to be learned still.
Well, this might be a good transition for Scott, because one thing I found of particular interest for me is in addition to the primary endpoints, the secondary endpoint with the lowering of LDL cholesterol. And it really begs the question, if the patients who had hit the primary endpoint, what % of them specifically had the secondary endpoint as well? And it begs the question, is this a synergistic effect? And what other secondary endpoints should we be monitoring and assessing? So I know that this was top-line data, but I'm really interested to see what comes out of this from that perspective.
So in addition to all the exciting implications we've talked about on the clinical side, for me it's very interesting in terms of the mechanism of action of this drug, because one of the unanswered questions is lowering fat alone good enough, number one, good enough to reduce fibrosis and get hard endpoints? And number two, does it matter how you lower fat? And the answer to number one, of course, still is not completely known, but it moves us towards a conclusion that reducing fat may be a good thing. I say that because, to my knowledge, the drug has no direct antifibrotic effect. And so any benefit in fibrosis and NASH resolution was largely, if not exclusively, based on its metabolic impact on hepatocytes and the reduction in the signals that drive inflammation and fibrosis. So I think clearly more studies to be done. With regard to the second question, I actually still would not assume that that any way you lower fat is the same. Because, there are drugs that lower fat that probably are not going to be as effective, even though the net result of them is that the fat lowering is consistent. So it may well be that lowering fat is necessary but not sufficient. You have to lower fat and create the right metabolic milieu or reduce the production of molecules that drive inflammation and fibrosis. And that may not be true from every mechanism in which fat is reduced. So again, lots of scientific questions here to tackle as well.
the critics to the approve as just to the approach, as Scott just said, was always said, it's not enough to improve lipids. Statins do that, no effect, and here we are today seeing NASH resolution and fibrosis regression. So I echo Scott's comment. We want to drill down on the link of, and I think there are a lot of links in the literature, lipotoxicity, activation of stellate cells, and all the work Scott's been doing over the years. So it seems to read out in a rough clinical trials. So that is fascinating.
And also, along the way, when their phase 2 study came out, they did not meet fibrosis improvement, and obviously the study was not powered to do so. But along the road, we started seeing NITs from their other MAESTRO-NAFL data and others showing that the stiffness measurements are improving. MRE was one of them, MAST was one of them, transelastography was one of them. So it's really good here to see that the NIT started predicting all along that fibrosis improvement prior to have adequately powered study. Just adding to the evidence that NITs biologically make a lot of sense because they represent a physiological process.
Yeah, speaking of drilling down, one other implication of this is to go back, and we all agree we should move past biopsy, but for as long as we have biopsy, let's start applying or expanding our efforts to apply digital pathology and artificial intelligence to see if there are patterns or subtle changes that were unmistakably present in the responders early on, or that there are other features that both give us a hint into new biology, but also define elements of the biopsy that may be more subtle than what pathologists can identify in further classifying or refining our assessment of the disease histologically. I mean, there's no question in my mind that any of the digital methods that are currently being developed are far more sensitive and informative than what a pathologist can tell us.
So 2 things on that. First of all, spoiler alert, Stephen, when talking about what's going to be presented at NASH Tag, talked about at least one presentation that will demonstrate for one drug the ability to use real simple liquid CBC level metrics to tell you relatively quickly whether that one drug is working or not. So I totally agree with Scott. We need to be able to figure out faster what doesn't work. Some of that might be very complex. Some of it might be simpler than we'd ever imagine. But yeah, it's something we need to get after immediately. And speaking of simple, I thought the most interesting thing about selecting LDL out of all the secondary endpoints to be the one to focus on today had nothing to do with anything we're talking about right now, but had to do with messaging the investment community that if the problems with OCA were pruritus and LDL, there's no pruritus here, and LDL, in fact, is a positive, not a negative. So going back not to the science of it, but to all the directions in which this message is out, I thought that was a really interesting decision. And by the way, the fact that we are now spending so much time talking about LDL makes that decision look really good because it integrates it into how people think about the profile of the drug. And I think that's a big step forward for how we broadly conceptualize what we can expect to see with drugs in this class. It's really interesting, all the different levels on which this stuff operates.
Yeah, the comparison with HCC is interesting, but I think as Mazen mentioned before, we have to remind that this is not going to be standard of care until we have full approval. So that's, I think, in the way you design your trials. Of course, you want to think about, if you start now, when are you going to be reporting out, and what's that going to mean? But for all the ongoing trials, and also the second thing, as Scott mentioned rightfully, you got to show efficacy for each drug explored in a combination in monotherapy versus placebo. So there will be more monotherapy, I think.
I have a question for the group, just wondering as we look forward now. So now there's a new bar. And we talked about that 26% wasn't really there yet. So do you think that combo therapy is going to be the next one before another monotherapy comes out that's better?
I think the example we can look to is actually from the liver cancer or the cancer community, where a drug is approved, then it's compared for non-inferiority to a second drug. At the same time, the next generation is take the first drug that's effective and add on a new drug with a divergent or complementary mechanism. And I would imagine we're not done with phase, with single drug therapies at all. But it's almost inevitable that given the response rate of 24%, that we're going to be rapidly, and I'm sure more than anyone, Madrigal will be very interested in amping up the effectiveness of their drug by adding a second agent. I'm sure there'll be a lot of recruiters who are auditioning for that role.
Yeah, that does not preclude the company, as Scott suggested, from looking for a combination for added therapy, because they're expected to be approved and they just want to build on that momentum and keep going. He's right. I think there will be multiple drugs tested to see if they're efficacious before they started to be added on to Resmetirom. But if I'm Resmetirom or Madrigal, I will probably now look into combining with a GLP-1, combining with SGL2s. I also want to remind the listeners that there were actually people on GLP-1 in this study. There were, yes, about 14 to 15%, and so as SGL2. So the Resmetirom seems to add to that in such a group, or probably not. We need to see the analysis, but still used, yes. But it will, I think even the sub-analysis will be interesting, but probably not enough numbers. But eventually, I would love to see it with systemic drugs or systemic drugs with an effect on the liver, such as the dual GLP glucagons, and adding Resmetirom, because you want to think about it as the first approved or one of the two first approved therapies for NASH.
Can I ask, and I know hindsight is a beautiful thing, Novo Nordisk and semaglutide, obviously approved in diabetes and obesity medicine, did they miss a trick by actually not really using non-invasive technologies for their liver health, given the high-risk populations that they used, and liraglutide, where they could have been collecting data for these studies? Or is it something that they could do protocol deviations for now, or protocol amendments, not deviations, to put into a later phase study within the populations that they've got? Because obviously it's going through the NASH, but obviously there's NAFLD populations that they're treating who would have had NASH. And the recent semaglutide patients that I've seen have all got perfectly healthy livers on FibroScan, and I know that's only one assessment.
I think they have a transient elastography in their study, not in the entire population. At the end of the day, they are getting NASH patients with biopsy-proven fibrosis. So I would say I would love to have more NITs, not just because I'm an IT person and I like NITs. I would like eventually to see a treatment response and pushing the field forward. But semaglutide has incredible efficacy as well as systemic effects and incredible advantage. To answer your question, maybe having more NITs is better, but are there disadvantages? I don't think so. It's a very efficacious drug. I would love, as a doctor and some patient advocate, I would love to see semaglutide with Resmetirom combined. There will be, before people starting doing big phase 2s and phase 3s, there are investigator-initiated studies and smaller studies proof of concept. So hopefully the small parties of combo drugs or small study, investigator-initiated study, few centers will start occurring in this space, especially what Roger said. There's tremendous excitement today, and I've consulted for companies that they intentionally did not want to go to NASH because they said the investors are tired and fatigued and frustrated. So yes, like sponsoring is important, and to me, like I would like them to pour some of that money in patient advocacy, awareness, in addition to just investments and stuff like that.
So a couple of questions or naive comments or both. Question/naive comment number one. I made the statement on the podcast several months ago that you could not get a combination drug approved until the individual elements have been approved. And we got feedback that isn't accurate in antivirals and in some situations in oncology. So it seems to me that it's certainly not reliable in those cases. It's not clear to me whether that'll turn out to be the case in NASH or not. I mention that because I think it's Terns whose strategy is about going straight to combination. And I know THR beta is one of the things they're looking at to go straight to combination in, and I doubt, So we may actually see people pursuing that path as a way to market. Thought number one. Thought number two, going back to something Scott said earlier, in combination with what Mazen just said. So SEMA has a 72-week trial that shows effect on NASH activity score, but not on regression of fibrosis. I think that is the single thing that
walks me right next to where Scott was when he talked about how compelling it was that this drug proved it could do both, or that it could regress fibrosis without having direct anti-fibrotic activity, because it says that there's something different going on here than what goes on with at least that GLP-1.
You're right. I think also Scott was commenting about the type of the fat that needs to be melted as steatohepatitis. I thought it was a brilliant comment that depends on which type of fat, and you aren't added to that lipotoxicity versus not. So yeah, I do agree with that, and I do agree that it's encouraging data for semaglutide. We should recall the data from the NASH CRN that showed clearly that if you reverse NASH resolution or you achieve NASH resolution or 2-point NASH improvement, you will eventually get fibrosis improvement. It's just a matter of time. So yeah, all great comments.
Good comment. And it feeds back to the theme of, personalized therapy, and obviously the presence of lipotoxicity as a driver of the disease to be, addressable by the mechanism of action of the drug that's required. Still, as you said, it's rightful that that trial has started recruiting in the US, and Europe is going to be starting up soon. So that is important. One more thing I had on my mind we haven't discussed yet. I found interesting that there's actually a second compound in class. So we're discussing out-of-class combinations, but there's a second thyroid hormone receptor beta agonist, and it's gotten a little more quiet around it. I think the company is called Vikins, and what could that mean for other? Do we need something in the same class to be developed, and see how those data are replicated, being different drugs? And go ahead.
Good point, Jörn. I think Terns is actually at great advantage of the results today. They have multiple drugs in the pipeline. They have TH-beta, they have FXRs and others. I do think that, sure, we would like to see more drugs and see their efficacy and all this. We still don't know if other TH-betas will achieve what Resmetirom achieved today. I do encourage other research in that area, and that incredibly important receptor that proven to play a big role in NASH.
Yeah, there's one other element that you embedded in your question, Roger, which is, or maybe you didn't, but I inferred it, is what about the unmet need in cirrhosis? Because to me, that's really the most urgent unmet need, because those are patients who are rapidly moving on to transplant lists or developing liver cancer. And yet they lose many of the features that seem so ripe for benefit from resmetirom, like reduction in fat, improvement in glucose homeostasis. Many of those patients are already losing weight. And so, correct me if I'm wrong, Mazen or Jörn or Kris, Madrigal has a cirrhosis trial ongoing. Personally, I wouldn't be surprised or disappointed if it wasn't nearly as effective in cirrhotic patients as in patients who have more full NASH with fat and inflammation, as much as I hope it is effective, because it may well be that there are different therapeutic targets that are critical to try to improve fibrosis once the liver has already gone to a cirrhotic stage.
I think you will find that it's easier to get investment now into other THR betas, because at least you've now proven that one should be able to get over the finish line. If there's a second one being studied and the entrepreneurs or a company, Viking being a small company, can make a compelling case that theirs might work better on any dimension that matters, that's easier money to raise than what everybody's trying to do for modes of action that haven't been demonstrated yet, even if they can prove staggeringly good results.
Yeah, well, there's a long history of first in class is not best in class. It started with statins, or at least that's the most vivid example, but it also worked in hepatitis B and C antiviral therapy.
And that's only because you live in hepatology, Scott. I will tell you that the only time in medical history outside of oncology, maybe even in oncology, up until 2015, that the first drug in was the market leader on the day it went generic was Prozac. And that's across all cardiology, lots of things. Just someone always came up with something better. Rachel, anything that you're working on, looking on right now that has any bearing on the question of what might be under the hood here? And what subtleties might be embedded in the questions people have been asking? And you can say no, by the way. That's a wild card question.
I would say yes. And actually, we're at an interesting point internally in the company, because, of course, we need to have for context for everybody on this call and all the listeners, my company is developing 2 non-invasive diagnostic tools. one that can differentiate steatosis from NASH, and a second tool that can stage fibrosis from a targeted epigenetic sequencing assessment. So the first question is linking each of those to biological relevance. What's happening directly in the liver? How do we know that this is coming from the tissue of origin and associated with a biologically relevant assessment? And for context of use, we initially started developing these tools as diagnostic focuses, but then it's something that within our pipeline we're looking at if we can also monitor therapeutic efficacy and which drugs and how relevant are these to specific therapies. So that is something that we are grappling with internally and something that we're really interested in conducting. So we're talking to some places right now that I'll keep internal at this point, but this is definitely relevant. And as a geneticist by training and as a focus who has a focus in genetics, a lot of this is really underexplored territory, and I think we're just starting to understand. So one thing is we were just discussing, first in class is not always best in class. So what we're doing is we're trying to get our MVP to market and then further develop that to make sure it is the best. So a lot of synergies here with what we're doing as well.
We're almost an hour into this, which is usually the point in time at which we start trying to move towards the end. But before I even think about moving towards the end, all of you, what itch haven't we scratched that you folks came out of today's announcement with?
One of the most important downstream benefits will be if these results are consistent over time from a standpoint of non-invasive tests, since we have such a rich plethora of non-invasive tests. And this is a trial where the non-invasive tests done in parallel that includes both MR as well as transient elastography and liquid markers, if you will, show a relationship between dose and response and dose and effect. Then I think that will also be a very important shot in the arm for those of us who believe that although liver biopsy is a valuable tool, is not ultimately how we're going to be helping this get into the hands of patients. And so the companion diagnostics used here from a treatment standpoint, from a point of view of payers, are going to be very critical. And I think we need to look at not only dose response effects, but also non-responder data, so that we can determine early on which patients are unlikely to benefit and then move on to other therapies. So I think the non-invasive testing showing in parallel similar results is very encouraging, and I think we need to really dive into that a lot more.
I'm glad that Kris mentioned this. I was, as you know, before I presented data from Resmetirom on something I really worked on for the last 2-3 years, and which is the MAS score, which consists of PDFF, MRE, and AST. And in a previous oral from the Resmetirom, we showed that MAS dropped with the Resmetirom compared to the placebo. So I'm anxious and curious to see if that MAS score will predict response to therapy. So I look forward to see that.
Roger, something that resonated with me also was in the follow-up questions by the investors, whether this wouldn't be now the right drug for all patients F1 through F4, opening this up. And the rightful answer was, of course, this trial was conducted in an F2, F3 population. And this is the label how it's going to read out. And I think we got to, with all enthusiasm, and I felt that enthusiasm in that question, why don't we give it to all, be reminded to really look at the data, be reminded on the patients that were enrolled. And I mentioned the metabolic risk factors. And that's important if we want to achieve those response rates as they have been reported. So while I could understood that person's question, why not give it to everybody with NASH, it's important to be remembered that the efficacy was shown within that inclusion-exclusion criteria, and we're not ready to just leave that yet.
I think, by the way, some of that was a confusion on the part of the questioner about the idea that 5% of that population was F1. And Becky Taub then actually explained why an F1C
patient might have been included. So I think, first of all, you and I are in violent agreement on this, and my guess is so is everybody else on this call. But I think that small set of F1 patients might have teased an investment analyst in a direction they shouldn't have gone. And that was my read on what that was.
Can I just give you one more point? If we take this and it gets approval, and then we look at the long term, this could just be one of the biggest anti-cancer drugs to be developed. Because NAFLD and NASH and the cancer-driving mechanism of non-cirrhotic NASH driving the cancer and liver cancer outbreaks at the moment around the world, this has the potential to turn around to be the biggest cancer investment that we could make by treating these patients. So if this really is as good as we're making it out to be today, we haven't even touched on the potential of reversal of liver cancer for these patients in the future. And that is just momentous.
Much more to say there, but I'll climb up in the interest of time. But a great point.
Well, actually, I just wanted everybody's closing comments. So I'm going to treat that as Louise's. And then, Scott, if you would like that to be yours, go ahead.
Just dig deeper, find the science behind this, explain why this drug worked and in whom it worked.
Mazen, if you got to jump, go next.
Congratulations to Madrigal. Thanks for what they have done. Congratulations to the patients, most importantly. Thanks to all those that underwent liver biopsy. We promise you we'll take this away one day, and look forward to see the future with the combination therapy and additional efficacies.
Yeah, an important milestone in terms of now we have 2 trials that have shown positive results in phase 3 and a trial that showed both NASH resolution and fibrosis improvement with a challenging regulatory pathway with regard to liver biopsy. So I think it in some ways reinforces that drug development in NASH is possible, can be done properly. This is going to lift all boats. I think it's very encouraging for patients, given that we have this huge epidemic that's already upon us in terms of fatty liver disease and the downstream consequences.
I just want to say that Stephen Harrison's picture of the graveyard, I'm so tired of seeing that negativity that this is finally an opportunity. Madrigal, congratulations, because your tombstone will not be added to that graveyard, and that's awesome.
So, the article that drove the graveyard might have been authored by a good friend and colleague of our podcast, Dr. Schattenberg, but we're going to save that thought for Professor Schattenberg. We're going to save that thought for another day. Okay, Rachel.
Well, I want to say thank you to the patients who participated in this clinical trial as well as various others. Thank you to Madrigal. And I just want to echo Louise's comment that this might be the biggest anti-cancer approach that we will have in for decades to come as a field. So I'm excited to see additional endpoints from a field perspective to further extrapolate the implications that this has. So thank you for the opportunity.
And just thinking of, all the investigators that are also contributing to the patients. I mean, this means you keep your patient in a phase 3 trial, even if you're not the highest specialized NASH center, only running NASH trials in that trial for such a long time, which is essential for those positive results. Congratulations to all these patients that stuck to it. And we're going to be reminded that it's an ongoing blinded study, and we're looking at the top-line data here, and it's just a big excitement from my end to learn and grow with that data.
Louise, you have one more?
No, I agree with my colleagues, fellow surfers, new to the surfboards, old surfers, you name it. The future's looking bright, and I did a hepatitis C one. It's not necessarily the first wave that you're on. It may be the second, the third, or the fourth wave that you catch that makes it the best wave.
In this case, 52 weeks on the way to 54 months. But so I come out of politics in the first place, most of you know. Deep Throat said to Woodward and Bernstein, follow the money. And because patients were generous with their livers and investigators had the faith to pursue, we're now to the point where I think we can stimulate, as Kris points out correctly, the second trial that had efficacy, significant efficacy in phase 3. We're now to the point where we can stimulate enough investigators to create enough money to, as Scott said, follow the science. There are systems that are self-amplifying, and you need moments when the system amplifies. The rules change today. Just a very simple level. The rules change. Stephen described this day as a watershed. I thought Yasmin Rahimi's question on the podcast this morning, where she said when she read the press release, it brought tears to her eyes, because God knows she invested in a lot of companies that didn't get there, or had her people invest in a lot of companies that didn't get there, or might never get there.
all the rules change for all of us today. And so I think it's just a banner day for all. Thank you so much for making time to be with us this afternoon. I will be back with a business report for our listeners in just a minute. And everybody take care. Thanks. Bye-bye now.
And now for an abbreviated Season 3, Episode 60 business report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. More on this tomorrow.
Still 3 episodes left, with only one alive recording.
So this is what happens when you cover a big story in real time. Stephen Harrison's NASH 2023 preview episode is delayed once more until tomorrow or Thursday. And it's a little shorter on NASH because it starts with Stephen's perspective on Maestro-NASH. After the NASH preview, we'll have 2 more episodes of interviews. The first of these, dropping Monday, December 26th, will feature Mazen, Noureddin, Ken Kusi, and Jeff Lazarus, with individual conversations on Tuesday and Wednesday. The final episode of the year will drop Thursday, December 29th, featuring Stephen and the 2 previously mentioned mystery guests now identified as Lain Al-Khoury and Ian Rowe. And with that, I'm off to enjoy the holiday. Thanks to the crew, Jake, who will be working through the holidays on this, Magic Mike, who had been working this week from Hawaii before the weather intervened, and Eric and Steve for all you do. And to our listeners, who matter most to us, until next time, stay safe, surf on. We'll see you on the podcast. Bye-bye now.
Please join us again tomorrow evening when Stephen Harrison joins Yarn, Louise, and Roger to preview the upcoming NASH 2023 conference here on the Surfing the NASH Tsunami podcast.